enthusiasm. Of course, Dr. Thayumanavan is a man of the soil. In that sense, he is really proud to be part of this conference, sharing his own experience and knowledge with us. So I'd like to thank Dr. Thay, he's fondly called Thay. I thank him for this, for participating and enlightening our students with a sense of pride and devotion to our students' community. A word of introduction about the person, Dr. Thayumanavan. From his own words, I am speaking. Dr. Thayumanavan is a distinguished professor in the Department of Chemistry and in the Department of Biomedical Engineering at the University of Massachusetts, Amherst. He is also the director of Center for Bioactive Delivery at the Institute of Applied Life Sciences at the University of Massachusetts. He received his BSc and MSc degrees from the American India. He received his PhD from the University of Illinois at Urbana-Champaign in 1996. Following a postdoctoral stint at Caltech, California, he started his independent career at Tulane University in 1999 and moved over to University of Massachusetts Amherst in 2003. The Thayumanavan lab is interested in translating novel polymer-based molecular assemblies into innovative materials to make a lasting impact in biomedical and sustainability applications. The group develops fundamental design guidelines for self-assembled systems that predictably respond to specific cues. These guidelines are used in enabling applications in a variety of areas, including self-healing materials, autonomously functioning systems, drug delivery, intracellular delivery of biologics, and cryptic catalysis. His work has resulted in more than 250 peer-reviewed publications in high-impact journals. The innovation in his work has also produced nearly 20 patents and patent applications. Dr. Thayumanavan has received numerous national and international recognitions for his creative contributions, including the Chemical Research Society of India Medal. He has also launched an early-stage startup company based on technology developed in his laboratory to solve complications based on liver diseases. And a few words about the topic itself he is going to talk to us today. It's titled Inspirations from Nature, Responsive Materials for the Modern World. And he has given a small abstract that will enlighten us on the nature of the topic he is going to discuss today. Nature has designed complex molecular and supramolecular networks of interactions to offer systems that bridge molecular scale initial events to macroscopic final outputs. Thus, Mother Nature has evolved intricate networks of systems that can bridge chemical events across multiple length scales and time scales. Design know-hows for generating artificial systems with such capabilities are primitive at best. We are inspired by this challenge. As a part of addressing this grand challenge, we have developed molecular designs that afford tunable host guest characteristics in supramolecular assemblies specific response to a variety of stimuli. Such a capability endows these assemblies with functional capabilities that could find use in diverse applications. In this presentation, he'll be focusing our discussions on supramolecular assemblies that are designed to respond to an atypical stimulus or feature atypical responsive characteristics. So with these few words of introduction to Dr. Thayumanavan's discussion today, I welcome him to the August gathering, online gathering internationally, and wish him all the best and hope that his talk will inspire many of our students to aspire his path and not confined to local place. Our students also will follow his path of reaching out to the horizons of the earth. Thank you very much. Thank you. Now I call upon Dr. Thayumanavan to address us. Welcome, sir.
Thank you. Thank you very much, Dr. Prakasam, for your kind introduction. So let me share my screen. So it looks like I need to share the entire screen. Can you see my screen?
Yeah, sir. Visible, sir.
So you're seeing only my slide, right? You're seeing my slides.
Yes, slides, sir. You can maximize slide. Yes, sir. Yes, sir.
You're seeing my title slide, right?
Yes, title.
Okay, thank you. Thank you very much once again for the invitation. It's a pleasure to be here. I am not, of course, with this setup, I'm not able to see any of the cameras. So if you have any questions, please let me know. And I'm very happy to talk about any questions that you have. So today, as Dr. Prakasam said, that we are focusing on inspiring students. So I'm going to really tell you a very simple project that we are doing. And it is more focused on how we think about a problem and how we arrive at solutions. And so I'm just going to give you one or two examples, depending on the time, one, maybe two examples. But I'm going to start with the inspiration. So the inspiration from nature. But before we go to the inspiration from nature, I wanted to look at something that all of us, including the younger generation, are very fond of, and that is our phone. And we all have our touch phones. And I told you in my inaugural thought process that you need to think about not only what are you looking at, it's a phone, how does it work? When you touch, the screen responds. How does it work? And why does it work a certain way? And if you start asking those questions, you learn a lot about the science, right? So, but let's think about, if you ask those types of questions and if you create those capabilities, how can we impact life as we know it? So if you look at the four objects around you here, okay, the one on the left top used to be a telephone exchange. You don't see telephone exchanges like this anymore. This was even my grandfather's time, right? And that was a camera during my father's time. That was a clock that you may still see in your home. And this may be a calculator that you might still see in some shops. But in the phone that we have here, we have all these capabilities. The phone does it even better than any of these together. We can hold it in our hand, and it can do so much more, so much more. Okay, and that is because there are so many innovations that has happened to create a system like this. So we are going to think about it. But I'm going to focus on just one topic, and that is touch. When you touch, something responds. did this also happen in nature? So you would have seen, I mean, I grew up in Tirunelveli and Madurai. Tirunelveli and Madurai, I have seen a lot of this plant. This plant is called touch me not plant, or it's called Thotta Chiningi, right? Thotta is Siningu, that plant, right? So this Thotta Chiningi plant, if you think about how does it, what is happening is when a person touches the edge of this leaf, the whole stem, all the leaves shrink, okay? The same way, this is called a Venus flytrap, and this leaf usually catches small insects, but in this case, it is dramatic that it even catches a frog. And these plants are carnivorous. They actually eat the frog. They eat the flies. And I'm also going to tell you a little bit about this particular shark. If you look at the shark, it is coming from the back. And if you look at the fish in the front, in the foreground, that fish is called hagfish. When the shark bites this fish, okay, and if you closely watch the mouth of the shark cannot close its jaw anymore because the hagfish deploys a gel in the mouth of the shark in such a way that it is not able to breathe anymore, so it doesn't completely attack the shark, the fish. So it's actually a self-defense mechanism of the fish. Similarly, this is another plant. When this insect goes and touches the mouth of this plant, there is a negative pressure that is created in this plant that actually sucks the insect like a vacuum, and it just pulls it inside and completely kills the insect. Okay, so if you look at all these systems, what you see is every system is responding to a touch. And so it's touch responsive system, much like the phone that you have. And can we learn from nature and make even more sophisticated systems than what you see in the phone? Those are the type of questions that we are interested in asking. So the thing about the thing about for example, this Thotta plant, the touch me not plant. If you look at the touch by that person, we can see it. And the response, where the leaves are shrinking, we can see it. But in reality, there are a lot of things that are happening in between the touch and the response. And that is molecular scale. Actual molecules, there's chemistry happening in between. In fact, there are thousands of chemical reactions that happen in between to have the macroscopic input come out as macroscopic output, okay, in nature. So, how can 1,000 reactions happen in such a short time? As soon as you touch, that reaction happens. How can thousands of reactions really happen? You might ask that question. Well, the reason is molecular scale processes happens at nanosecond time scale. Okay, very, very, very quick, extremely quick. Molecular scale processes can happen. But macroscopic processes, where that move a stone, that's macroscopic process, those can happen in seconds and maybe milliseconds, but not faster than that, right? So you can imagine you can bridge thousands of molecular scale processes to bring about one specific macroscopic process. And one of the things that we are interested in my group is how do we make this bridge between molecular scale processes and macroscopic processes? And why are we interested in this? Because I asked you to, so I'm asking the how. I told you what we are doing. I'm asking you how we are thinking about how we can bridge this gap. And next question is, why would we want to do this? Why are we interested in doing this? So for that, let's think about responsive systems in general. So in a responsive system, we need two things. You need an input, that is a touch, is the input, right? And the output could be a fish deploying a gel, or the leaves shrinking, or that plant sucking that insect inside using a negative pressure. Okay, that could be the output. But for a chemical system, if you're interested in generating chemical systems, the input, we can classify them into a chemical input, or it could be a physical input, or it could be a biological input. So the touch responsive system belongs to the category of mechanical input. It's a mechano input. And if you go to a COVID test and they do the swab, and there are now COVID tests are available as color change. You can swab your nose and there's a color change, and that tells you whether there is COVID virus, and that is response to a pathogen. Okay, in your lab, you might have used pH paper, and that's a response to the pH. So there are responsive systems that you can create, and you can broadly classify into these chemical, physical, or biological inputs. But the really interesting thing about the why happens if you think about what can be the output, even if you think about different outputs. Okay, so let's say the output is a transduction event, meaning there is a change in a color or change in the fluorescence or something in a material. And if there's a change, we can use this for sensing. Okay, or the output could be a molecular release event where you have a capsule that encapsulates molecules, and we can release that molecule in response to that. Maybe that's your output, and we can use it for interesting purposes, and we'll talk about that also. Or let's say there is a change in a material going from, let's say, hydrophilic to hydrophobic, and how can we use that material? So that's a physical property change. So there are all these variations that are possible. So if there is a signal change, if there is a transduction event, you can imagine using it for sensing or diagnostics or theranostics. We'll talk about each of these in a second, okay? And you can use it for self-healing and drug delivery if it's a molecular release event. And if it's a physical property change, you can use it for self-cleaning materials as an example. But let's actually go through each of these outputs in a little bit more detail so that you can imagine applications that are not possible. So for each of these outputs, and there are more, we are working on even more outputs like this in our group, but I'm just going to focus on these three just to illustrate to you what are the possible uses that we could have with these systems, okay? So first, let's think about transduction event. So some of you might know this, and some of you may not. But there is a home pregnancy test. If a woman wants to know if she is pregnant, they can actually use this home pregnancy test. That you can check with a color change from urine, right at home in their bathroom. Okay. And there's a lot of chemistry involved because there are chemicals released because in a woman when they are pregnant, and because of that, and if you can sense those chemicals and change color because of that specific chemical, you could tell whether somebody's pregnant or not. Or let's say we want to detect if somebody has a cancer and where is the location of that cancer. And if it is deep inside, and if it is not big enough, can we still image the cancer? And if the cells are distributed all over the body, would we be able to image it? We can detect it using the sensing and the diagnostics tool because we have a signal change that is specific to cancer. Similarly, let's say we have a molecular release event. For the same patient, we have diagnosed the cancer, but would we be able to deliver a molecule to a specific location and release it only in that location? Because cancer drugs are toxic. They can kill all cells. And if you want to selectively kill cancer cells, can we make those nanoparticles or objects that we create go to the cancer cells and open up and release the drug only there and not anywhere else? Therefore, we eradicate the tumor, the cancer, but the person lives, okay, and does not have side effects. Or for self-healing materials, imagine somebody is flying. In the mid-flight, there's a crack in the wing. That's very scary. That's very dangerous, right? But if we coat this wing with a self-healing material, when the mechanical force of a crack happens, we release a healing agent where it automatically heals the wing. Then, at least until the plane lands, that plane can be very safe. So those advanced materials, we can imagine if we create chemistries like this. In fact, the first step towards that has already been created, not by our lab, but by another lab which works in the same field, where they have self-healing paint, where if you paint your wall with the paint and if you scratch, if the kids go and scratch with some sharp object, the paint automatically heals. So it's one step closer to the dream that we can have here. Similarly, if you want to have a physical property change, so this person walking in watch the rain drops on the shirt, on the jacket that they're wearing. The raindrops are not, they're rolling off because the surface is hydrophobic and it doesn't penetrate the clothing. But you can imagine if you were to wear these clothes before the rain, and if it is really, if it doesn't even allow water exchange, water inside, that person will also suffocate and they will sweat a lot, which is not very comfortable. So can we design the raincoat in such a way that the shirt is fairly open so you'd exchange, it's like a cotton shirt, that you exchange air and moisture very well. But then when it becomes wet, when it raindrops touches it beyond a certain wetness, it'll become hydrophobic and it'll automatically shut down and it won't let water through. Okay, it's a common man's use. The same way, if we have soldiers who go into dangerous battlefields, and if they are dealing with chemical weapons or biological weapons that they might come into contact, can they wear protective clothing in such a way chemicals their skin, so they are protected, but normally the air and the water can go through, so they can normally walk and they don't feel too hot. Okay, so there are hundreds of applications like this we could think about as to why we want to do this. But we need to understand fundamentally how to connect these inputs with these types of outputs. And that's what we are interested in doing in my research group. So from a chemistry perspective, the currency of chemistry is actually to think about correlating structure and function. Okay, so that's what we are going to try to do, to develop new structures so that we can create new functions and so that we can go to new materials. That's what we're interested in. Okay, so I'll come back to this idea of the touch me not plant, but I will actually more specifically come to this hagfish. So I told you about this hagfish. This is the one that is getting bitten by the shark in the background. And you can see that fish in the front, you can see it also here. And what happens is remember when the shark bites, I told you that it deploys a gel. And even in a very soft way, this person touches this fish very softly, gives this mechanical stress to the skin of this fish. And you could see how much gel it makes, and the person can pull it out, so much of the gel. Okay, so it's a defense mechanism. And if you do it hard, it'll make a lot of gel to protect itself from shark attack. So what we asked is, how does this thing work? And can we create artificial material, not based on touch, but based on some other input? Can we create a material like this if you use light as the input? Okay, so I'm going to take you through the chemistry as to how we came about doing this. So first, we want to understand the mechanism. How did hagfish do it? So it turns out in hagfish, it has a compartment. Okay, maybe we'll call it a vesicle. And there is a vesicle compartment in which it has these, the vesicles inside, and it also has these threads. But those threads and these contents of inside these vesicles, what is called, there is a protein called mucin. That mucin protein and these threads cannot see each other, even though they are in the same fluid. Okay, but when you squeeze it, these vesicles, these little vesicles burst. And when they burst, they release these proteins. And when they come together, they make this gel. So this is what biologists found out about how hagfish is making this gel rapidly. So can we learn from this? And can we design materials that does something similar? So what we were interested in is we encapsulate a protein, and we have other materials. So this is, you can think about it as like the thread, yeah, around other molecules that are around. And these molecules themselves don't make a gel, okay? So they can coexist because the enzyme is inside the vesicle, the protein or the enzyme. Okay, I'm going to use it exchangeably. Enzymes are proteins. So they're inside, so they can never react with this material, so it's not going to make this gel. But as soon as I open this up using light or any other source, would I be able to make this gel? That's the question that we asked. Okay. So how do we do it? How do we want to do it? So the mechanism by which we thought of doing this is, first let's generate the vesicles itself. So we want, remember, in order for us to do this, we need to break this vesicle, break this container using light. So how do we do that? What is the design? So what we came up with was two polymers, and these two polymers are going to interact with each other. And they're going to, when they interact with each other, they're going to make this vesicular compartment. That is the wall of this. Okay? And the reason why this interaction is very strong is because it is called what is called polyvalent interaction. So you have so let's say this red color here is a negative charge, and the plus here is a positive charge, and this negative charge and the positive charge like each other, and they are going to interact with each other. But what is more important is the strength of interaction is strong in water when it is polyvalent, meaning when a polymer interacts with a polymer. If I have only a single anion interacting with each of these, so I have one here, one here, one here, but they're independent of each other, they're not attached to each other, the strength is not that strong, okay? So what we are going to do is we are going to start out with having multiple contact between these polymers, and then we are going to use light to break it up in such a way that the interaction becomes a single interaction, not multiple interactions, not polyvalent, but monovalent. And that will cause this to break up and open up. And we're going to see whether that happens. And if you want to understand polyvalency versus multivalency, think about this. You might have seen shoe straps where you have this Velcro, right? Or many other things where you put Velcro with each other. And if you look at the contact points between Velcro, the reason Velcro is strong is because you have multiple contacts from both sides. So you can think about it as negative and positive charge. But if you take a very small piece and if you make a single contact with this, you can easily take it off, no problem. But when you have a multivalent contact, it becomes very, very difficult to take it off. And that's what we are using, changing the polyvalent contact to the multivalent contact. And that's what we're going to design, okay? So how are we going to, what is the molecular design criteria? So then we are going to encapsulate our proteins inside. And then when we break it up, it will open up, okay? So let's now think about the molecular design in order for us to get there. So so we talked about this idea of breaking polyvalent contact to multivalent contact. So we have two polymers interacting with each other. What we want to do is to convert one of the polymers to not being a polymer. A polymer is something that we have multiple copies of, okay, that is attached to each other. And if I break it up, I can make it into individual components. And so if I have poly negative charge, I can have individual negative charge. And this will interact with a polycation, poly positive charge, much better. But this one will be much weaker because they're monovalent. Okay? So we are going to do it in such a way that when I shine light, I break this here, break the chemical bond here, and that is going to cause this whole molecule to fall apart. And we're going to design the chemistry to do that. Okay? So let's see whether that happens or not. So if I'm able to do it, I can do this gelation. Okay? So what is the molecular design? And here's the molecule. So if you look at this molecule here, okay, I have this whole system, and here is the polymer, and in the polymer I have this negative charge. Okay, you can see this negative charge. This is the negative charge that we are going to use. Okay, and when I put this within brackets, what that means is there are multiple copies of this negative charge along the polymer, just like this. I'm showing you 3 here, but in reality, you can have 10 or 100 or whatever. Okay, and then at the end of this polymer, where I have this blue star, I have this nitrobenzyl compound, this molecule. The reason I have this molecule is because, well, first of all, when I interact with a positive charge, I'm going to use this polymer, and that polymer, this polymer, the breakable bond, it's just going to be a polycation, and it's going to make this vesicle. But when I break up the negatively charged polymer, it's going to open up and release the enzyme, and that is going to cause gelation. So that's what we're going to see. Okay, so let's see if that works or not. So how does the mechanism work? So I have this polymer, and I told you that this is the one that is responsive to light. So what happens is when I shine light, this bond breaks here. This bond between the carbon and the oxygen breaks, and that's a known photochemistry. I'll be happy to tell you why that happens later if you're interested. But when that breaks, what you create is a negative charge here, or just a carbon, this functional group here. But this functional group is unstable because it can lose carbon dioxide very quickly. You can see CO2 here, and that CO2 can be lost. And if you lose the CO2, okay, there is a cascade reaction that happens continuously throughout the polymer that ends up in generating completely this monomer. So this polymer with n number of units completely breaks up into individual monomer chain, where this negative charge on the side chain is not polypolymer, but it is a single unit. Okay, and this adds to water, and you make this molecule. So if this R is a hydroxy group, or if it is a negative charge, this will be a negatively charged individual molecule, but we started out with a polymer. So that's the way this reaction works. So just to tell you that, does this really work? You can focus on this particular peak that corresponds to this. And as you shine light and you watch that signal keeps going away. But while that signal goes away, as we shine light for a longer time, other signals come up. And you can see these different colors for different parts of the molecule in the product that comes up that shows that the way we design this reaction is really the way this is working. Okay, this is just a polymer. We haven't done the vesicle and the sheet yet. So if I take this polymer and if I just put it in water without light, the polymer is very stable. That's the red line. But if I shine light, the polymer degrades with time. Okay, so now once we made those polymer, remember I said that we can mix these two polymers and we can make these vesicles. Well, does it really happen? Do we really make these vesicles? I have shown you in drawing, but do I have the data to show that it is really making these nanometer sized particle? So here is the image. This is a transmission electron microscopy. You can see the size of this bar, it's about 100 nanometers. That means the size of these particles is about 100 nanometers or 200 nanometers diameter, okay? So we can make this, and we can even zoom in to see if we can make these wall and the inside is water. And we have encapsulated proteins and enzymes inside here, okay? So now what happens is when you shine light, are we able to release the system? And so we have characterized this using a fluorophore. So here we have encapsulated not an enzyme, but a fluorophore. When you release the fluorophore into water, we will have the fluorescence increase. And in fact, we see that, okay? When we shine light, the fluorescence increases. When we don't shine light, there is no change in the fluorescence. Then can we do the experiment that we said, which is, can we use an enzyme that we encapsulate inside, and then when we release it, that this becomes a gel, okay? And in fact, we have done that experiment. So what I'm showing you here is we have a bottle, a vial with the solution that we have before we shine light. And when we invert it, the solution, of course, flows because it's a solution. It just comes down because of gravity, right? But after we shine light, we make a gel. And you can see the gel is staying on the top because the gel doesn't flow as fast. If you wait for a long time, maybe it'll come down. But it doesn't flow very quickly, just like water will. And so that shows that it makes a gel. So we also do what is called rheology. And if you're an engineer, you will understand rheology, but I won't go through that. But you do the rheology to show that this is really a gel. That this thing is really a gel. And so, in a sense, what I have shown you is that by using a molecular scale system, by learning from nature about how hagfish operates, we can design systems in which gelation happens. And we have done, literally tens or hundreds of systems like this that are responsive to different inputs. And I just give you very, very simple example for you to understand how we think about molecular design. And I will leave you with one other system that is a little bit more complicated, so I won't go through the details too much in terms of the molecular design, but, but a completely different way of thinking about it, where it is inspired by, more than it is inspired by nature, we took on a challenge because a company was interested in a challenge, and I want to tell you how we developed the chemistry for that, okay? So this company Pepsi, which many of you might know, came to us. And I don't know if in India this has become popular, but in America what happens is when people drink Pepsi type of drinks in restaurants, and I'm sure in India also this has become in McDonald's and things like that, and KFC and things like that, you would have seen it. The cup, you go to a cup and you put ice here, and then you pick whatever drink you want, and if you press this lever, you will get the drink. The drink itself is at room temperature, and it becomes cold drink only because you put the ice, okay? Now, what they're interested in is the flavor. So these Pepsi, when they are produced in some industry, some factory, and then they go to the stores, the lifetime is about 6 weeks. After 6 weeks, the flavor starts deteriorating, okay? So what they want to do is the flavor that causes you to feel that you are drinking something good. These are not real, right? These are flavor molecules that just makes you feel good because of the smell and the taste. They want to encapsulate it in such a way that it doesn't degrade when it is being transported and produced and transported at room temperature, at normal temperature. But when it hits the ice, you release the molecule. So you feel like it's a fresh orange juice or fresh orange soda or whatever, right? But it is really not, but you just make it feel that way. And so this is really an interesting problem, because what they're really asking is, can we make a process happen at a lower temperature, even though the process does not happen at a higher temperature? That is really counterintuitive. That's opposite of what you normally think about, because you may remember your Arrhenius equation. Arrhenius equation says that a rate of a process is directly proportional to temperature. So at higher temperatures, the reactions go faster. At lower temperatures, the reactions go slower. So how can we make things go faster at lower temperatures? It is counterintuitive. So we developed a process called, we actually didn't develop or discover, we actually, I want to say, we exploited a process called lower critical solution temperature process, where there are molecules out there that are soluble at lower temperature becomes insoluble at higher temperatures. So these insoluble things can be more static. They are not as dynamic, but the soluble things can become more dynamic. And can we take advantage of this system to create what this company is looking for, is a question that we asked. And in fact, we were able to create such a system. And I will just, in the interest of time, I will just show you the final result here. And that is, these molecular release does not happen at higher temperatures. So I'm showing you 25 degrees Celsius. But at 10 degrees Celsius, this molecular release happens much faster. And at intermediate temperatures, you can tune the release. So only at lower temperatures, the molecular release happens. So this could be used for what Pepsi is interested in. But remember, the molecules that we make for this process are not food grade product. That means you cannot add to food. These we don't know if it is safe. But the process that the understanding that we provided is something that, industrial chemists can use to hopefully make these types of systems that can become a lot more dynamic. Okay, so again, I won't go through the details of this. I will stop there. And so this is my research group. This was an old photograph, but this was my research group a couple of years ago, I think. We have a lot of students, and I'm very proud to say that my group is almost 50% women, 50% men, and I do it intentionally because I want people of all gender, all countries, all religion, whatever background you come from, you can succeed, and poor, rich, none of those should matter, and you can succeed together. And I'm very particular about bringing that diversity. And I have people in this group from almost all states in India, China, Turkey, Thailand. I'm just looking at their photographs to see what other countries I'm seeing. Of course, the US, Germany, Mongolia, all kinds of countries. Okay. Of research interesting, and there are so many more research areas that one could think about. And I hope you pursue research as a future for yourself and in higher education, and do great things for our society. Thank you very much.
Thank you, Dr. Thayumanavan, for the very interesting discussion on the topic of how we can learn a lot of things from nature itself to create similar processes artificially so that it can benefit the humanity. More than your topic itself, the concluding remark regarding your sense of social justice, that you want to have inclusive growth for everyone in your own lab as well as in all that you deal with. We are all really proud of you. I think that is a very good message for our students, that opportunities are available, that people like you are able to encourage people at the lower strata to come up in life. So that's a very good message for us, because our institutions, a Jesuit college like ours, is standing for social justice and inclusive growth of our students. So it's very interesting that you carry on this good work outside your own country. It gives a lot of satisfaction and pride of belonging to the same region where we all come from. Thank you, Dr. Thayumanavan, and it's open to the audience to clarify or propose any new ideas based on what you have listened to him.
Students, I particularly want to encourage you to ask questions, because the first step in being creative is inquiry. Never feel, even if you don't ask me questions today, that's fine, but I hope you do. But always be willing to ask questions, because I always tell my students in the first class of my first time I teach every year, every time I teach a class for the first time, I tell the students, ask me a lot of questions, and there is no such thing as a stupid question. And in fact, there is only one question that is really stupid, and that is a question that you had in your mind, but you did not ask. If you had a question, but you did not ask, that's a stupid question. As soon as you ask, that is not a stupid question anymore. So please do ask questions, and I'm happy to answer. And I will also say, when I presented to high school students here, because I also do what is called outreach to inspire students in high school in the US as well as in India. And when I present to them, one of the things that I ask is, like Dr. Pragasam said, can you tell me what I should be doing with responsive materials from your perspective? What is your need? What is an application that I have not thought about? So I'll give you a funny example. A girl in a school in the US told me, Professor, can you make self-healing pants? Because she said every high school student has at some point or the other has felt that their pants or skirt or something situation, and they were worried about it and they wanted to change. But if you just, instead of self-healing the airplane wings, can you self-heal our shirts and clothes so that we will never have the tearing off problem? I thought it was very creative, right? It's a very creative problem. It's not going to be a product for other reasons, for economic reasons, but it's a really creative idea. So please ask away. Somebody raised a hand. Yes, please go ahead.
Martin. Professor Martin wants to ask a question. No. I think professors also can ask. It's open to students primarily, but professors need not hesitate to ask any clarification. Certainly. I have a couple of questions, Doctor. One is about sportsmen. We were talking about self-healing. we see a lot of cricketers, when they get hurt, the doctor comes and sprays something or other. Are they self-healing ointments, or it also takes a longer time to heal? Or is it instantaneous healing?
Yeah, self-healing bones or self-healing biological systems is probably 50 years away. If we have self-healing airplanes that are 10 or 20 years away, I think self-healing biological systems is probably about 50 years or more away, because biology is inherently so complex. So if you want to implant something, people will not like to put anything that they don't need. First of all, right? So any foreign object.
Yeah.
But remember, the self-healing system is something that we have inbuilt in us. But bone breakage, for example, that self-healing does not exist in our immune system, or even it does, but it is too difficult, especially as we become older. So self-healing systems there are a lot more complicated. So it's probably, it'll take many decades before we realize that, but that's something the younger generation can start thinking about.
Yeah, the second question is a very age-old question. Why we are not able to imitate photosynthesis? It's also a natural process that's occurring all around us, yet we are not able to exactly reproduce and solve human problems, food and so on.
So yeah, photosynthesis in primitive ways we have mimicked photosynthesis. So you can look at even the light-induced chemical reaction that I did. In a primitive way, you can look at it as photosynthesis because you're using light to execute the chemical process.
Yeah.
But nature has made extremely complex systems. As I said, it can weave 1,000 processes into a process to convert carbon dioxide all the way to carbohydrates, to sugars and leads and things like that. That process, there are people who are working on that also. It's called CO2 fixation, it's carbon dioxide fixation using light. And so that's also something that people are working on it, but it's a stepwise process. If you look at 20 years ago, we were much poorer in mimicking photosynthesis than we are today, but we have a long way to go. Nature, the excuse that scientists give, I think, is reasonable. Nature had billion years to optimize its process. And we are trying to optimize the process in our lifetime of 50 years or 60 years. So it's difficult, but we are slowly evolving.
Very good. Thank you, doctor.
Yeah, thank you.
Yeah.
Thank you, everyone, for having me. It was a pleasure presenting this conference.
So, thank you, doctor, once more, and I ask the organizers to take over from me. Thank you.
Thank you.
So, thank you, Father, and Dr. Thayumanavan, sir, for deliberating this wonderful session. Now, the session, we will go for the break, and we will come back at 11:45 a.m. Thank you.
Thank you. Thank you.
Amal sir, what's the time? Come back time?
Sir, 11:45 we will come back.
11:45.
Yes, sir.
Okay. Sir, Mutramalingam, sir, kindly note, sir. For us, for Indian time, we have to come back after the break, 11:45. Okay, sir.
Okay,
Kindly
sir.
note, sir.
Okay, sir.
According
Thank
to
you.
that, you arrange, sir. Thank you, sir.
Thank you, sir.
அப்பா one second, இதான் live stream start பண்ணணும், end stream.